Hello everyone and welcome back to AP Bio with AP Playground. Today we're reviewing Unit 3, Cellular Energetics. Unit 3 consisted of 7 subunits, many about enzymes but also some about cellular energy, photosynthesis, cellular respiration, and then molecular fitness. So what you need to know for cellular energetics: what are enzymes and how do they work, the environmental effects on enzymes, endergonic versus exergonic reactions, ATP, photosynthesis, cellular respiration and their steps, fermentation, and types of molecular fitness. Okay, here we have 3.1, enzymes. So enzymes are biological catalysts, and catalysts change the rate of reactions, and are also not used up in the process of the reaction. So enzymes are reusable and they're biological catalysts. The majority of them are proteins, except for ribozymes, which are made out of RNA, and as a result they have protein folding structure consisting of primary, secondary, tertiary, and possibly quaternary structure. Now they greatly speed up reactions because by changing the rate of reaction, making it faster, more reactions are able to take place. And most enzyme processes are reversible, and oftentimes if there's an enzyme that breaks apart a certain product to split it into two reactants, another enzyme will take those reactants and put that product back together again. It's all about maintaining a balance of what's needed in the cell. And lastly, enzymes are very, very specific. There is going to be a specific substrate for each enzyme active site. Now, substrate is whatever an enzyme is acting upon. And it can change the substrate, it can combine two substrates, or it can separate. And they're bound to the active site of the enzyme, which is the part of the enzyme that will be acting upon the substrate. And this forms an enzyme-substrate complex. So if you look at the top here, we see there's a substrate entering the enzyme active site, and it's got a specific shape. The substrate is made to specifically lock in with the shape of the enzyme active site. And once the enzyme-substrate complex has been formed, the substrate is then separated into two products, which are then released from the enzyme. As you can see, the enzyme is not used up in the process and then can be reused to break apart that same substrate, and usually only that substrate. Now, how enzymes actually change the rate of reaction is by lowering the activation energy, which is the amount of energy needed to start a reaction. And we'll go into how they do that a little bit later. But within uncatalyzed reaction pathway, meaning no enzymes have been used, there needs to be some energy put into the reaction, this is the activation energy, to then perform the reaction and get the product. But with an enzyme, the amount of activation energy needed is a lot less, meaning that you can, with the same amount of energy, perform a lot more reactions, and you're getting the same exact product. Now let's take a look at the induced fit model. So what is the induced fit model? The induced fit model is the way scientists explain how enzymes actually do lower the activation energy. And the induced fit model says that the substrate and active site have matching shapes and attracting charges, and when the substrate is attracted into the active site, the enzyme makes a conformational change to make them completely fit. So as you can see here at the top, the substrate almost fits the enzyme active site, but not perfectly. However, once the substrate has entered the active site and you get that enzyme-substrate complex, the enzyme makes a conformational change, a change to the shape of its active site that allows it to fit perfectly with the substrate. Then the substrate is broken apart into products or put together, and the products leave the enzyme. And the way this lowers the activation energy is by pressing upon the bond, making stress or forcing two substrates together. So in a catabolic reaction, or a reaction where a molecule is being broken and energy is being released, the conformational change of the enzyme places stress upon the bond, lowering the activation energy. This conformational change is going to place stress upon the bond that's going to separate the substrate into the two products, helping speed along the process. Versus an anabolic reaction where molecules are being made, and this is going to consume and store energy, conformational change forces the two substrates together, lowering the activation energy because it's easier to form bonds when they're so close together. Now, the amount of energy produced or consumed through the catabolic and anabolic reactions doesn't change. However, the activation energy is lowered, so you're able to perform more reactions with the same amount of energy. Moving along, it's time to talk about environmental impacts on enzyme function. Now, enzymes can be denatured, meaning the tertiary and quaternary structures of the enzyme are broken apart. The bonds in them are broken apart due to their fragility. And this happens once the enzyme is placed in an environment that is outside of its optimal range of pH and temperature. And this is the reason for compartmentalization, or where each certain process has its sort of own part in the cell. It's maybe enclosed by a membrane, or it's located within a place that has those specific optimal ranges, allowing it to perform its function. Now, sometimes the denaturation is reversible, but other times it also isn't, which is why enzymes are kept in their optimal range of pH and temperature. Now, you can look here at this graph. You see this is the temperature graph. Now, each enzyme has an optimal range. You see this is the optimal temperature for a typical human enzyme, around 35-ish degrees Celsius. And as you can see, there's sort of going to be a sloping increase of enzyme efficiency, the rate of reaction, as you reach that optimal range. And the reason for this is the molecules are going to move around faster as the temperature increases, and they're going to move around more, allowing them to bump into each other and more substrates to enter the active site of the enzyme and be acted upon. However, once you leave this optimal range and you start going too far past the optimal temperature, the enzyme will become denatured. Similarly, for pH, this is comparing pepsin and trypsin. Pepsin is going to be in a much more acidic pH. It's going to function at around a pH of 2, while trypsin will function at a pH of 8. And there's sort of a slow increase into that rate and a slow decrease, and outside of this range, the enzyme won't function at all. In this portion of the range, you have some functionality, but it's not going to be optimal like it is up here, which is why it's so important for the enzyme to be in its optimal range and to be compartmentalized. Now, additionally, changes in environment such as the increase of the amount of enzymes or the increase of the amount of substrate concentration are going to result in a change in the amount of product over time. So as you can see here, we have our base reaction. Over this period of time, you're going to get this moles of product, and at the start, when the reaction begins, all of the substrates are going to be filling up the active site, so you see sort of a steep increase in product. However, over time it'll level out once all enzymes are filled and once substrate has run out and there's no more product being made. Now, if you increase the enzyme concentration, that means you're able to fill more active sites with substrates initially, allowing you to get a sort of steeper increase and reach the same point, the same amount of product at a faster rate, because you're able to fill each enzyme faster. Now, if you have a substrate concentration with the original amount of enzymes, that means your rate of creating product, your rate of filling the active sites with substrates isn't going to change. You're going to continue on the same rate. However, your ending amount of product is going to end up being larger because you have more substrate concentration, you have more substrate to act upon. So instead of, let's say, 17 moles of product, you're going to be able to go up to 20. Moving along with 3.3, it's time to touch upon cofactors and coenzymes. So cofactors are non-protein helpers that bind to the active site, and these are metal ions such as zinc, iron, and copper, while coenzymes are cofactors that are organic, and they also bind to the active site. And the most common example would be vitamins. So as you can see here, you've got your enzyme and you have your cofactor or coenzyme binding to the active site, which is allowing the substrate to then be attracted into the active site and fit correctly. However, if this cofactor coenzyme wasn't there, the hexagonal substrate would not be able to enter the active site of the enzyme, and the enzyme would not be able to create that conformational change. Finishing up 3.3, we have to talk about inhibition. First off, competitive inhibitors. These are molecules that compete with the substrate for the active site on the enzyme, and they have the same shape as substrates, so they're able to enter the active site and prevent a substrate from entering. And these can be irreversible, such as poisons. Now, there's also non-competitive inhibition, and these are molecules that are going to impede with enzyme activity by binding another part of the enzyme, not the active site. Usually they bind to what's called the allosteric site, and this can cause a change in the shape of the active site, preventing the substrate from entering the active site of the enzyme. So if you look down here, we have a competitive inhibitor where the inhibitor is located inside the active site, preventing the substrate from entering the enzyme, versus a non-competitive inhibitor where the inhibitor is attached to the allosteric site, which changes the shape of the active site, preventing the substrate from entering. Now, not all inhibition is bad. Feedback inhibition is when the end product of an enzymatic pathway can switch off the pathway by binding to the allosteric site of an enzyme in the pathway. Meaning that if you have object A and then goes through the enzyme, becomes product B, goes through enzyme 2, becomes product C, which is what the cell wanted, once there's enough of product C within the cell, C will allosterically bind to enzyme 1, preventing the whole process from starting. That way, once there's enough of the product within the cell will stop producing more, and it'll only produce what it needs because this is efficient and saves energy. So an allosteric site can both inhibit and stimulate enzymatic activity. Moving on to thermodynamics and free energy, topic 3.4. So metabolism is the totalities of an organism's chemical reactions. How much energy is inputted into the organism and then it expels. Now, energy is the ability to do work, and life requires a constant input of energy. Once you're out of energy, you're dead. Now, thermodynamics is the study of energy transfers in matter, and the first law of thermodynamics says energy cannot be created or destroyed. So although life does require a constant input of energy, this is because the energy is constantly changing or being transferred, not because it is being destroyed. The second law of thermodynamics says that every reaction reduces the amount of available usable energy, most of which is lost as heat, which is this energy transfer that requires then you needing to input more energy into your body. It can also be called the law of entropy, saying that energy will quote-unquote diffuse, or there will be quote-unquote disorder. Energy is always going to move from a place of high energy to low energy, because moving in the reverse is going to take up more energy. Now, free energy is the part of a system's energy that is able to perform work, meaning it can cause reactions, meaning it can do things. And a change in free energy is notated by delta G, delta meaning change, and G standing for Gibbs, who came up with this concept of Gibbs free energy. Now, in an endergonic reaction where something is being produced, this can also be called an anabolic reaction. Energy is stored through the formation of a bond, and free energy is going to be consumed. So as you can see here, we have an endergonic reaction, and we have the inputs by themselves, the reactants by themselves, plus the input of an energy, the activation energy, is going to result in products that have more energy than the initial reactants. And when the reaction took place, some of the energy was lost and some was stored. So as you can see here, you have your activation energy. You start with your reactants at nearly no energy. You give it energy, you use up energy, some of it's lost, and the remainder of it is going to be stored. This is energy absorbed. This is going to be the energy of products, which is going to have more energy than the initial starting reactants, which means it's going to be a positive change in free energy. The product has more energy than the reactants. Meanwhile, in an exergonic or catabolic reaction, energy is going to be released through the breaking of bonds. And this is going to cause a change that is negative in free energy. So the reactants already have energy stored within them, and some energy is used to break apart the bonds, but more energy is released than it took to break apart the bonds. You see, we have this great amount of energy released, and the energy of the product is going to be a lot less than the energy of the reactants because all of the energy has been released, meaning you have a negative change in free energy. The product has less energy than in the reactants. And enzymes assist with the transfer of energy by lowering the activation energy and allowing for the separation of anabolic and catabolic processes, and oftentimes similar processes are located together. So one anabolic pathway might be located next to a catabolic pathway because oftentimes the reactants for one are going to be the products for the other. Now it is necessary to discuss ATP, adenosine triphosphate, the currency of the cell and the primary source of energy for cells. Now ATP is a nucleic acid, meaning it consists of a ribose sugar, a nitrogenous base, and phosphate groups, and it has the adenine nitrogenous base and 3 phosphate groups, with the third group being especially unstable and joined to the other two by a very high energy bond. And work in the cell is done through the hydrolysis of ATP, meaning the addition of water to break apart bonds, and energy released in the exergonic reaction, forming ADP. So this energy comes from the breakage of this high energy bond, which results in ADP adenosine diphosphate with these 2 phosphate groups as well as 1 inorganic phosphate. And this energy release can be used for an endergonic reaction, meaning energy coupling, where the energy for one exergonic reaction, energy release, can be used to make something with an endergonic reaction. So as you can see here, once ATP has been hydrolyzed, you've got energy for cellular work, energy that can be used for endergonic energy consuming processes, and you're left with your ADP plus your inorganic phosphate group. Now, they are rejoined back into ATP from energy from other catabolic exergonic energy releasing processes, which then restart the cycle. So energy is being inputted from energy releasing processes to hydrolyze ATP and create the energy for cellular work, endergonic energy consuming processes, making ATP truly the currency of the cell that allows work to happen. Now we need to begin discussing photosynthesis, and to do that we'll start with autotrophs versus heterotrophs. Autotrophs are producers of their own food. They produce food for themselves, such as plants, whereas heterotrophs are consumers. They are dependent on autotrophs for both food and oxygen, because plants also produce the oxygen in the atmosphere. And this evolved from the cyanobacteria and early photosynthetic organisms that created oxygen through performing photosynthesis, and then later joined eukaryotic cells and became chloroplasts through endosymbiosis, now resulting in the chloroplast double membrane. And the chloroplast is going to be the site of photosynthesis in the plant cell. Animals do not have chloroplasts, and animals cannot perform photosynthesis. And the equation for photosynthesis here is going to be 6CO2, so carbon dioxide, plus 6H2O, water, plus light energy. It's going to result in glucose and oxygen. Now, essentially, all photosynthesis is the conversion of light energy into chemical or food energy in the form of glucose. And photosynthesis is an oxidation reduction reaction, otherwise known as a redox reaction, meaning that there are going to be loss of electrons and gaining of electrons within the reaction. So as you can see here, the carbon is going to be reduced. It is going to gain hydrogens, and along with gaining hydrogens, protons is going to gain the electrons that come with the hydrogen atoms, whereas the water is going to become oxidized. It is going to lose reactions, it's going to lose its hydrogens. Now let's take a look at the chloroplast in greater detail. The overall inner gel of the chloroplast is known as the stroma, and the chloroplast is filled with these membrane sacs known as thylakoids. And the membranes of thylakoids are going to be used to establish gradients that we'll touch upon later. And thylakoids contain pigment that absorbs light known as chlorophyll. And chlorophyll is what is going to make the leaf green, because although it absorbs most wavelengths of light, it reflects green. And lastly, stomata are holes in the leaf through which carbon dioxide can enter and oxygen and water vapor leave, meaning that this is going to be the exchange for all of the gases needed for photosynthesis. And thylakoids contain chlorophyll. Photosynthesis consists of 2 parts, light-dependent and light-independent reactions. The light-dependent reactions are going to convert solar energy into ATP, which is then going to be used to create glucose in the light-independent reactions. And light-dependent reactions are going to start in photosystem 2, also known as P680. That's the type of chlorophyll it's got in there. And what's going to happen is photosystems are just where photons or light energy are absorbed by pigments. And once light hits the photosystem 2, it's going to result in an electron placed into a higher energy state. This electron is going to jump around the light harvesting compound until it reaches the reaction center and later the primary electron acceptor of the photosystem. And this primary electron acceptor is then going to send the electron down the electron transport chain, which we'll talk about in a minute. Now, to replace this electron, water is used, where the electron from the water molecule replaces the electron in photosystem 2, oxygen leaves the cell as a waste product, and the hydrogen atoms are then entering the thylakoid space on the other side of the thylakoid membrane. So, we're now made it to the ETC, where electrons are passing between photosystems through a cytochrome complex of transmembrane proteins. And this is going to then result in chemiosmosis, where hydrogen ions or protons are being pumped into the thylakoid space by the energy the passage of the electron creates. So protons are being pumped into the thylakoid space, but they are unable to create equilibrium because now there's a lot in the thylakoid space and not as much on the other side of the membrane. Equilibrium is not attainable because they are ions and have a charge. So the only way they're able to diffuse across the membrane is to then pass through the transmembrane protein ATP synthase. And as the hydrogen ions pass through ATP synthase, this is going to power the enzyme, and it's going to attach a phosphate group to ADP when activated, creating ATP. So through this ETC, we formed ATP, and this is a process known as chemiosmosis. Lastly, simultaneously, light has been hitting photosystem 1, and the same thing is happening where an electron has been raised to a higher energy state. It's jumping around. It eventually reaches its primary electron acceptor and gets sent down another electron transport chain, and the electron lost from PS1 is going to be replaced by the one from PS2, while, as you recall, the one from PS2 is being replaced by the breakdown of water. Now, this last electron that's been sent down this very last electron transport chain from PS1 is going to enter the electron carrier NADP+, leading to the creation of the full carrier NADPH, which is then going to be used in the light-independent reactions along with the ATP formed through the chemiosmosis. So now we must discuss the light-independent reactions. This is going to happen in the stroma of the chloroplast, and we've taken our ATP and NADPH from the light-dependent reactions, and light is not needed for this part, as well as CO2 from the atmosphere, which passed through the stomata, and it's going to enter the Calvin cycle. So 3 carbon dioxide are going to enter the Calvin cycle, and they're fixed, meaning incorporated into an organic compound, to this 5-carbon sugar RUBP through the use of an enzyme Rubisco. So as you can see here, to the RUBP, carbon dioxide has been added. Now, the ATP and NADPH used that we gained from the light-dependent reactions are going to be used, the ATP phosphorylating the compound, and electrons are added through NADPH. The remaining ADP and NADP+ will then go back into the light-dependent reactions to be reused. And the Calvin cycle is going to form G3P molecules. And one is going to leave the cycle, while the other 5 rearrange themselves and reused in the cycle as RUBP. And obviously you need 2 G3P molecules to create 1 molecule of glucose, as glucose is a 6-carbon sugar. And to create 1 molecule of glucose, there must be 6 turns of the cycle to fix 6 molecules of CO2, as within each turn of the cycle, there's going to be 1 addition of a carbon to the RUBP. Moving along into cellular respiration 3.6, we're going to be discussing aerobic respiration and also anaerobic respiration. First up, aerobic respiration is using oxygen, aerobic, turning macromolecules into ATP. And while glucose is the primary molecule and the only molecule we'll be talking about in this course, in terms of cellular respiration, other macromolecules can also enter the respiration cycle and create ATP from their food energy. And this exergonic release of energy from glucose is used to phosphorylate ADP, turning it into ATP, which can then be used as the currency of the cell for endergonic reactions. Just like photosynthesis, it's going to be a redox reaction, reduction oxidation, tied to losing and gaining electrons. And our cellular respiration equation is going to be glucose plus oxygen yields carbon dioxide plus water plus chemical energy. And it's nearly the same as photosynthesis, as the reverse of photosynthesis, except that you are gaining chemical energy instead of putting in light energy. So really, these reactions are coupled, and it's just a transfer of energy into another form. And as you can see here, the glucose is going to be oxidated or oxidized. It's losing hydrogens when it becomes the carbon dioxide, while the oxygen will become reduced. It's gaining hydrogens and as a result of that also gaining electrons. Here we're going to go over the steps of cellular respiration that are all substrate level phosphorylation before we get into the part that uses the oxygen, that's the aerobic part. So first off, glycolysis. This is going to be anaerobic and in the cytosol before you even enter the mitochondria. This suggests that it evolved earlier. This was the respiration process before there was oxygen in the atmosphere, before the creation of organelles. And your net gain is going to be 2 ATP, it takes 2 and gives 4, and it's going to fill 2 NAD plus, or reduce them, electron carriers, forming NADH, and it will split the 6-carbon glucose sugar into 2 3-carbon pyruvates. So we're now at 2 3-carbon pyruvates. And now we've moved into the mitochondrial matrix, and we're going to perform what's called the link reaction. A carbon dioxide is going to be removed from each pyruvate, so there we go, 1 carbon gone from each, and this is going to be exhaled. 2 more NADH are going to be created, and now we have 2 acetyl coenzyme A's. Lastly, we're entering the Krebs cycle, or the citric acid cycle, and it's going to take 2 turns for 1 glucose, because of course we have 2 acetyl coenzyme A's. All of the carbons are going to be lost as CO2. So first there's going to be 1 spin of the cycle for this acetyl coenzyme A, and there we go, carbons lost as carbon dioxide and exhaled, and then the same thing with the second one. So through this cycle, every single carbon of glucose has been emitted as carbon dioxide. Additionally, 2 ATP, 6 NADH, and 2 FADH2, which is the reduced version of the electron carrier FAD, are going to be created. Now, a good way to remember the electron carriers for cellular respiration, because they are not the same as the photosynthesis NADPH, is thinking NAD and FAD. You have NADH and FADH2. But it's important to remember that NADH is not the same as NADPH. Now, we've discussed all of these parts of cellular respiration, but we haven't seen the creation of much ATP. There were 2 from glycolysis and 2 from the Krebs cycle, but that really isn't much. And now we come to the part where the majority of the ATP is going to be created, in oxidative phosphorylation, meaning we're going to have phosphorylation of ADP using oxygen. And in the inner membrane of the mitochondria, because the mitochondria is a double membrane organelle due to endosymbiosis, there is going to be an ETC, an electron transport chain, just like photosynthesis, consisting of transmembrane proteins. And it will be powered by electrons from the carriers NADH and FADH2. And as the electrons are traveling between these proteins, there is going to be the pumping of hydrogen ions across the membrane from the matrix into the intermembrane space. And electrons are moving through the chain attracted to the final electron acceptor, the electronegative oxygen atom, which the combination of which is going to form water. And of course, oxygen is electronegative. We learned this back in 1.1. And because of this, there's going to be an attraction, there's going to be a pull that brings the electrons through the transport chain, and eventually is going to form water as a waste product. Now, these hydrogen ions in the intermembrane space form an electrochemical gradient, meaning firstly, there's going to be a gradient just because there are more of them on this side of the membrane than the other, but also there's going to be a differing charge. It's going to be a lot more positive up here in the intermembrane space. But they're unable to cross the membrane because they're ions. So in order to follow their concentration gradient, they must go through ATP synthase, just like photosynthesis. And once they travel through ATP synthase, you're going to get a phosphorylation of ADP into ATP. And this here is going to produce 34 ATP, which is by far a much larger amount than what you get from glycolysis or from the Krebs cycle. So this chemiosmosis, this oxidative phosphorylation, is going to be the part where you're making all of the ATP out of aerobic respiration. And obviously this is going to evolve because larger organisms need more energy, and this is made possible through the atmosphere created through photosynthesis. And of course the NAD plus and FAD are going to go back into glycolysis link reaction Krebs cycle to be reused. And the hydrogen atoms are now going to be back in the matrix, the ions. So cellular respiration is really just the inverse of photosynthesis. The equations are nearly the same inverse. One is going to produce oxygen while one consumes it, and one is going to produce carbon dioxide while the other consumes it. Now, there is a type of respiration that does not involve oxygen, and this is known as fermentation. And fermentation is going to be using glycolysis, which, as we already know, doesn't give much energy, but it's better than none. The only issue is that the NAD plus carriers, the NAD carriers, are going to become reduced with electrons, and once they're completely filled up, glycolysis is not, you won't be able to repeat glycolysis. So the whole point of fermentation is freeing up these electron carriers, oxidating them, so that you're able to repeat glycolysis. There's alcohol fermentation, where the pyruvate from glycolysis is converted to ethanol, carbon dioxide will be released, and then NADH will be oxidized. And this appears in things such as yeast, like beer and sourdough. And then there's lactic acid fermentation, which forms lactate as a waste product and also oxidizes NADH. This is going to form things like fermented vegetables and kefir. And it's also going to be used in humans, so when you're running, you get cramps. That's because you're out of breath. Cellular respiration cannot function with the amount of oxygen you have. So fermentation takes over, glycolysis happens, and there's a buildup of lactate, which is going to be resulting in you having cramps.